my video software and I hit stop, and so I ended up with a really short. But I do want to start up here with complement. Complement is a cascade of proteins that are resident in the bloodstream, and it is much like the clotting cascade. It is very complicated, and I don't want you to know the entire complement cascade. So I'm going to try to be as explicit as possible in terms of what you need to know. So this is a system that, it's called complement because it actually complements, or it runs sort of synergistically with immune reactions. And the complement system is about 26 different blood proteins. They start out as zymogens, and then they're activated through the complement cascade. And they will work to destroy bacteria and certain viruses, and they will also clean up the mess. Okay, the complement system actually will dissolve residue from the immune system fighting against pathogens. Okay. So the complement works by a cascade reaction, and a cascade reaction is just a sequential response where the first step activates the second step activates the third, and so on and so on and so on. And at each point, not only is the reaction sequential, but it's also amplified. So you start out with this very small stimulus, and then it's amplified over many, many reaction steps. So you get a much larger response. Okay, and there are three different pathways. And of course, my screen is cutting off the last one, but the first pathway is called the classical pathway. Second one is called the lectin pathway, and the third is called the alternative pathway. And each one of these pathways is initiated by a different type of factor. So if you break down the pathways, the classical pathway is just activated by the presence of any antibody bound to a microorganism. So if you have an antibody, this is the third line of defense, because you've got B-cell mediated immunity with antibodies. And if the antibody is bound to a microorganism, then the constant region of the antibody will be sticking out, and it will initiate the classical pathway. Okay, so once the antibody binds a microorganism, then the classical pathway is initiated. The lectin pathway is where you have a serum protein. This is just a host serum protein, and it binds mannan. Mannan is a sugar that you find primarily in the wall of fungi. And once this host serum protein binds mannan, then that will initiate the lectin pathway of complement. Okay, so this is more specific for fungus. And then you have an alternative pathway, and this is when the complement protein binds both the normal cell wall and the surface component of a microbe. Okay, so when you've got sort of a sandwich in between a normal cell wall, normal human cell wall, this is a microbe, normal cell wall of a microbe, the complement protein basically binds this, and that initiates the alternative pathway. So you should know the three things that initiate complement. You should know the classical pathway is activated when you have an antibody bound to a microorganism. The lectin pathway is when a serum protein binds mannan, and the alternative pathway is when a complement protein binds the normal cell wall and the cell surface component of a microbe. Okay, so make sure you know those three by definition. Okay, and this is how they work. The classical pathway is rapid and efficient, and you have circulating antibodies, IgG and IgM, and these will interact with the surface component of a microbe, and that initiates a C1 complex. And the C1 complex then cascades to C4, then C2, then C3, and then it goes through the common pathway, which involves C5, C6, C7, C8, and C9, and then the membrane attack complex. And the membrane attack complex basically pokes holes into the membrane of the foreign invader. The lectin pathway is initiated by mannins, and you've got a mannose binding lectin. That lectin is a serum protein that initiates C2 and C4, and then it goes into the common pathway. And then you have an alternative pathway, which is slower and less efficient. This is initiated by the bacterial or fungal cell wall. Viruses, virus coat proteins can do this, or surface of parasites like helminths. This is initiated through C3. And then you have other complement proteins called factor B, factor D, and properdin. And then that goes to the complex pathway. Now, I'm not that interested in you knowing these components. I would want you to know the three different pathways and what initiates them. But as far as the cascade, I'm just not going to make you memorize that. So, but first we have initiation. Okay, so that's initiated depending on which pathway you're talking about. Then as you move through the complement proteins being activated, then you have amplification and cascade. Then you have polymerization, and this is the polymerization involves the membrane attacks component, which creates a collar of different proteins. And I have a slide that shows this. And this polymerization then leads to membrane attack, and that can digest holes in the cell membrane of the pathogen. And by digesting holes in the pathogen, then the contents spill out. You actually kill the pathogen. Okay, and so here we have our pathogen. Okay, and you can see that the pathogen is activated directly by the presence of an antibody. Okay, the antibody then initiates the cascade through C1. It actually cleaves C1. And then it goes through different steps in the pathway where it goes through C2, C4, okay, can actually come down here and activate C3. Then this assembles to form an enzyme. This enzyme then will cleave C5. C5 becomes C5a and C5b, and then C5b then calls forward this entire polymer, and you see a sort of a collar at C9, and C5b, C6, C7, and C8 also form this membrane attack complex, and then this membrane attack complex then joins with the cell membrane, and when the cell membrane is eclipsed, then the cell starts to fall apart. Okay, water then comes in due to osmotic pressure, and then the pathogen bursts. Okay, very complex. There are a lot of different components of this particular pathway. This is a classical pathway because it's being initiated by the antigen and the antibody binding together, and then it's working through C1. But you see how C1 activates C2 and C4. This creates an enzyme that activates C3. This creates another enzyme that activates C5, and then you get the membrane attack complex. Okay, so this is just a classical pathway, and I don't, again, I'm not really interested in you knowing the details of these different pathways, but you should know what initiates the three different pathways of complement. Okay, then moving on, then we have iron-binding proteins and antimicrobial peptides. This is another way that you can fight against pathogens. Okay, and iron is generally not available in the body, which is a good thing. Pathogens need iron, so they're looking at scavenging iron from human tissues. Okay, and so this becomes a rate-limiting factor for the growth of pathogenic bacteria. So it's looking for things like transferrin or other proteins in order to give up its iron. And so iron-binding proteins like hemoglobin, transferrin, lactoferrin, and ferritin will actually take up iron, and they'll use iron for physiological reactions. Okay, and when they bind iron, during pathogen attack, then the pathogens can't get at the iron, and the pathogens don't survive. Okay. There are also antimicrobial peptides. These are a group of them is called the defensins, And they're very small, anywhere between 12 to 50 amino acids, which is the small size of a protein, all smaller than insulin. And they have the capability of inserting themselves into the prokaryotic plasma membrane. And when they do that, the membrane falls apart. And when the membrane falls apart, then the pathogens themselves die. Okay, so that concludes chapter 14, finally. And I'm going to pause this and then I'll move along to chapter 15. Okay, here we are on chapter 15. So we're going to talk about specific immunity and immunization. I'm going to give you my take on this. Wading in trepidation into the world of immunology, which is not my special place, but I want to cover it anyway. I think it's really important. Microbiology of infectious disease. So our objectives for this chapter are to discuss the third line of defense, immunity. Okay, so this is the acquired immune system that revolves around T cells and B cells. And we'll understand the steps in development of specific immunity, lymphocyte development, antigen presentation, B cell response, and T cell response, not really in that order. Okay, and then we'll discuss immunization, which is the process where the body becomes immune to a particular pathogen, and vaccination, which is the artificial means which can result in immunization. Okay, so specific immunity. This is the type of resistance that you get after you contract ailments in childhood, after you can contract ailments as an adult, and this provides long-term protection against future attacks. So once you've been exposed to something like measles or chickenpox, or even specific types of the flu, then you develop some type of resistance. So the next time that you see that particular pathogen, then your body has developed an immunity, and that's a part of your acquired immune system. Okay, it's adaptive. We can say it's adaptive. We can also say that it's acquired, but it is not innate. It doesn't. It's not something that you're born with. And it's a product of T and B lymphocytes. And it is part of what we call immunocompetence, which is the ability of your body to react with countless forms of the body is wonderfully made and has the ability to sense, detect many, many different combinations of antigens on the surface of pathogens and even internal to pathogens. And that makes you immunocompetent, so you can recognize these different patterns, and you can recognize what is self, you can recognize what is non-self, and you can also recognize what is commensal or beneficial. So your immune system will keep those types of microbes in check, but they will not completely attack. Okay, so antigens are molecules that stimulate a response by T and B cells. Okay, so antigens are located on or are associated with pathogens, and we can also call these immunogens. They stimulate the immune system, and so an antigen is the more typical term, but you can refer to them as immunogens as well. Usually these are proteins or polysaccharide molecules. Usually they have to be quite complex in order to elicit an immune response. And they can be on the surface or on the inside of cells and viruses. Okay. Antigens are generally foreign molecules. They're not self, and they generate an immune response. And they are very individualized, okay? And our immune repertoire has been designed to see these individualized proteins and polysaccharides, and they stimulate a specific immunity. So PAMs are not antigens. PAMs are pathogen-associated molecular patterns. That's part of innate immunity. And if you get anything out of this chapter, I want you to know the difference between antigens and PAMs. Antigens are acquired immunity. to CPAMPs. It's written within your innate immune system. The receptors that are resident in macrophages see PAMPs directly and bind PAMPs directly without having to come into contact with a PAMP in order to elicit an immune response. Antigens, however, you have to have an initial run-in with an antigen in order to elicit an acquired immune response. Okay. So in the specificity of immunity, let's step back. I don't like the way I said that. Acquired immunity is very selective. Okay, so the antibodies and T cell response that you get from the acquired immune system is highly selective. So you're looking at specific portions of antigens that antibodies and T cell receptors will bind onto, and that's what constitutes the acquired immune system. So it's highly specific. So in other words, if you're immune to chickenpox, that immunity will not extend to mumps. They're two different viruses. They have two different structures. And so therefore you would see that they're highly selective. And when you have specificity, not only do you have specific immunity to that particular pathogen, but you also have specificity to the antigen on that pathogen. So your immune response is based on what antigens have been seen previously. Okay, you also have memory. And memory means that after you have your initial response, your initial immune response, over long periods of time, your body will remember through memory T cells and memory B cells, and they'll be able to rapidly mobilize these lymphocytes after they have been previously programmed to recall their first engagement with the invader. So you have an initial engagement with the invader, and then after that you have memory cells that will rove the different compartments of your body, and they will remember that they saw this particular pathogen before, and then you'll have a much more rapid and robust immune response the second time you see that pathogen. Okay. And so we have lymphocyte development. Okay, so we have lymphocytes that are T cells and B cells. And then through phagocytic cells, we'll have presentation of antigens. Antigens are then presented to different types of T cells. Okay, they can also be presented, as we'll find out, to B cells as well. That's sort of a different mode of action. The antigens then are presented that will challenge T cells and B cells. B cells lead to antibody production, and T cells lead to cell-mediated immunity, things like cytotoxic T cells and natural killer cells. Okay, so back to B cells and T cells. We talked about this before. B cells mature in the bone marrow, and T cells mature in the thymus. Okay, and then both will separate out and will migrate to lymphoid organs. And they're constantly in circulating, so they're in the circulating system and in the lymphatic system. Okay. And in order to elicit a specific immune response, first you have antigen presentation. Okay, remember antigens are the molecules on pathogens. So we first have to somehow present those antigens in order to activate T cells and B cells. Okay. So pathogens, those are the bad guys. They carry antigens. Those are the cell surface molecules, and they attract macrophages. Macrophages are the Pac-Men that eat the bad guys. Macrophages ingest the antigen, and then they migrate to the nearest lymphoid organ. Okay, so once they've ingest the antigen and the pathogen as well, I should say that, and then they will migrate to a lymphoid organ like the spleen or the appendix or the tonsils or some type of lymphoid organ. And then they process and they present the antigens on their surface. Okay, macrophages actually decorate their surface with the antigens of the pathogen that they just ingested, and they present those antigens to T cells or T lymphocytes. Okay. Antigens are also available to activate B cells, and they require the assistance of T helper cells. Okay, and stuff is popping up on my screen. I am so sorry. Okay, so they activate B cells, but B cells require the assistance of T helper cells. So T helper cells actually get the process moving. Okay. Then B cells and T cells then proliferate and differentiate. Okay, so you get different clones of B cells and T cells that match onto the antigens that have been displayed by the macrophage. Okay. Some of the B cells and T cells that mature and proliferate are called memory cells, and these memory cells will ensure future immunization. Immunization is not vaccination. Immunization, these memory cells are a part of the process of the body becoming immune. That's what immunization is. If you get anything from this chapter, please know the difference between immunization and vaccination. Okay, so here is a nice busy slide for us to break down. So we have a garden variety lymphocyte that's out of lymphocyte class. And if it goes to the thymus, it becomes a T cell. If it goes to the bone marrow, it becomes a B cell. Okay? So these cell lines are just in weight. They migrate to lymphoid organs. Okay? They can go to the lymph node or the spleen or the appendix or the tonsils, which are lymphoid organs. And they just lie in wait. Okay? And then we have a phagocytic cell. Okay, that can be a macrophage, it can be a dendritic cell, it can be a neutrophil. These are dendritic cells. Okay. And these phagocytic cells, they're undergoing phagocytosis, and they have little markers on them, these little Y-shaped molecules. They're called MHC markers. MHC stands for major histocompatibility complex. Okay. And each one of these MHC molecules then will ingest an antigen, and then the antigen, or I'm sorry, each one of these dendritic cells or phagocytic cells will ingest the antigen, and then portions of the antigen are displayed on the MHC marker molecules. So the MHC markers are these red Ys, and the little green dot on top of the red Y is the displayed antigen. Okay? And then a T helper cell, which matches with that antigen, that has the same complementary shape on the T cell receptor, then this T helper cell will get stimulated, okay? And then the T helper cell will stimulate B cells, okay? So the T helper cell will stimulate B cells as well. Now, there's another way that you can stimulate B cells. Here's a B cell, and it's studied with antibodies. It's not studied with B cell receptors, it's studied with antibodies because it's a B cell. And you can also get little pieces of antigen that will stimulate this B cell as well. So this B cell, we call it a helper B cell, can be stimulated just by portions of the antigen that are floating around in the bloodstream, okay, or in the lymph system. So it can get stimulated on its own, but mostly what happens is that through this antigen display by the phagocytic cell, the T helper cell then will stimulate the B cell, and then that will cause the B cell immune system to activate. So you get an activated B cell, you get an activated T cell. Some of these T cells become memory cells. Okay, some of the B cells become memory cells, and these memory cells then just will proliferate over a lifetime, and they will remember that particular antigen that was displayed way back here. Okay, and they just rove the compartments, and they provide long-term immunization. Activated B cells will also make plasma cells, and the plasma cells each secrete antibodies. They could secrete different types of antibodies. Antibodies go into the circulatory system, and these antibodies then will bind directly to the pathogens. Okay, they recognize the antigens, and when they recognize the antigens, then they bind the pathogens directly. And then, of course, this is difficult to see, but here the activated T cell then becomes a T helper cell. Look at these T helper cell, which helps with the cytotoxic response. T regulatory cell, which regulates the response, and then a T cytotoxic cell, which is actually a killer cell. And the T cell system down here is cell-mediated immunity, and we'll talk about that further. And then over here, the B cell is antibody-mediated immunity. Lots of words on that slide. Okay, so let's talk about antibodies a little bit. Okay, B cells give rise primarily to plasma cells. We don't call plasma cells B cells, but they differentiate from B cells. And each plasma cell releases only one specific type of antibody to tissues and blood. The antibodies attach the antigen, and they'll target them for neutralization and destruction. Okay, and then we have three types of T cells. T cells are activated by antigens, and they produce these three types: helper T cells, and helper T cells will activate more macrophages. Helper T cells will assist B cell processes, and they also activate cytotoxic T cells, which will kill infected human cells. Then we have regulatory T cells that control the entire T cell response. And then we have cytotoxic T cells that will destroy infected host cells and also foreign cells. Okay, so remember, three types: helper T, regulatory T, and cytotoxic T cell. Okay. Now, in order for lymphocytes to develop, they have to have cell markers. Okay. And these markers will allow attachment to non-self or foreign antigens. They also bind cell surface receptors that indicate self. Okay, you don't want the immune system to attack self, so you have to have some type of binding with self in order for the immune system to decide what it's going to attack. And then these markers will receive chemical messages, usually by small proteins, to coordinate their response, and they aid in cellular development. Okay, and the main cell surface marker is what's called the major histocompatibility complex, or MHC. Okay, and MHC glycoproteins, glycose is sugar, so that's our sugars are protein. They're located on all cells except for red blood cells. These are also known as the human leukocyte antigen system, HLA. So sometimes you see MHC, sometimes you see HLA. And there are three classes. The first class is class 1 MHC, and that just defines self. Okay, all that does is that's just a marker on all nucleated cells except for red blood cells, and that defines self. So when you see class 1 MHC, then that just basically says I'm a human cell. Okay, then class 2 MHC is what appears on immune regulatory cells. Okay, specifically class 2 MHC is involved in display of antigens. Okay, and then finally you have class 3, and these are proteins that are involved in complement system, among others. We won't talk as much about MHC class 3. We're primarily concerned about class 1 and class 2. Okay, and here is class 1 MHC. It's just a multi-subunit protein here. Okay, and this identifies self. It's not designed in any way, shape, or form to, it can bind other proteins. It's got an antigen binding cleft, but it's not fashioned in order to display an antigen. Okay, and here's class 2 MHC. Okay, and this is found on some types of white blood cells, mainly phagocytic cells that are going to display antigen. And it's got a specific design here so it can display antigens directly and interact with T lymphocytes. Okay. B cell receptors, will directly bind to antigens, and B cell receptors are just antibodies that are studded on the surface of B cells. T cell receptors bind processed antigens, okay, via macrophages, and they complex with the MHC molecules on the antigen-presenting cell surface. Okay, so T cell receptors dock with the MHC class 2, and that docking is facilitated by a specific antigen that will guide in the T cell receptor. Okay. So when you have maturation of different lymphocytes, you have millions and millions of different types of B cells. Each distinct B cell line is specific to a distinct antibody structure, okay? And that antibody structure is delineated by the genetic sequence for the gene that's encoding that antibody, both the light chain and the heavy chain, okay? So when B cells mature, when they've basically been stimulated by the immune system, then you get genetic modification and selection, and each B cell will mature to make a specific type of antibody. And these adhere to specific binding molecules, and through the lifetime of that cell, they will contact a specific type of antigen. Okay, so they're very antigen specific. Each B cell, after it's matured, is specific to one type of antigen because it's only displaying one type of antibody. Okay, T cells have T cell receptors. They're much less complex than antibodies, and these T cell receptors will bind MHC class 2 plus the antigen, okay, and that helps the T cell to mature. You also have a co-receptor called CD3, and a CD3 co-receptor also involves binding of the major histocompatibility complex class 2. T cells also express CD4 and CD8 co-receptors. And a CD4 co-receptor binds MHC class 2. And a CD8 receptor you only find on cytotoxic T cells. Remember, cytotoxic T cells only attack, they will attack self. They will attack human cells. Okay, so they will actually bind MHC class 1 for infected cells. And in that case, the cytotoxic T cell will destroy self. Okay, it's not trying to kill you. It's basically trying to kill human cells that are infected to prevent the spread of viruses and bacteria. Now, in terms of what our immune system can do, this is so wonderful. We have a set of genes that is the human immune repertoire, and you've got basically over 500 gene segments. They mix and match to bind to specific millions and millions of different types of specific receptors for antigen. Okay. During B cell maturation and T cell maturation, the gene sequences are extensively rearranged, and so you get a high level of diversity. Each B cell is specific to one antigen. Each T cell is specific to one antigen. And then as these cell lines expand, then when we have a B cell line or a T cell line that is specific to one antigen, we call it a clone. Okay. And this clone, if we take a single clone and we grow it up and we just let the cell divide over time, then we'll have a monoclonal line of antibodies, okay, or a monoclonal line of T cell receptors. Okay, each clone makes a specific antibody or makes a specific T cell receptor, and it can only bind one type of antigen. And so by the time that T cells and the B cells have matured and they've reached lymphoid tissues, okay, each one is equipped, and it can only recognize a single unique antigen. So they lie in wait, okay, and the B cells and the T cells are only specific to one type of antigen, and once that antigen is displayed by the macrophage, then the T helper cell that corresponds with that antigen will bind, and then that will cause a clonal expansion of those B cells and T cells for that particular antigen. Okay. This is a type of antibody. This is a typical IgG antibody. I'll talk about the different types of antibodies in a few slides. Okay, and here we have light chains, which are smaller on either side. And then we have heavy chains that are larger and kinked in the middle. The heavy chains are held together by disulfide bonds. The light chains and heavy chains are also held together by disulfide bonds. And then you have a C region, which is in red, that's for constant. And then you have a variable region, a V region, which is in yellow, that's for variable. Okay, and at the end of the variable region for the antibodies, then you have these sites called antigen binding sites. Okay, and this little cleft in here on this side of the antibody, and then also on this side of the antibody, is specific to one type of antigen. Okay. If we look at the top of the antibody where the Y splits off, this is called an antigen binding fragment or a Fab. And if we look at the bottom here, this is called the constant region, sometimes it's called the complement activating region. Now, in order to build this, we have a variety of domains. We have a V domain, a G domain, a J domain, which is a joiner, and then C for constant. Okay. And these different domains, this is part of the genetic sequence here. They'll mix and match. You have variable region 1, 2, 3, 4, and so forth. D region 1, 2, 3, all the way to 12. Joiner region 1, 2, 3, 4, and then constant regions. And these will mix and match for heavy chain complex and a light chain complex. And when they mix and match, then they will provide precise binding. You've got such an immune diversity here. Once these antibodies fully assemble, you have millions and millions of combinations, and each one can bind a specific type of antigen. Okay, and this just shows the route of maturation. All cells, all these lymphocytes, the progenitor cells, lymphoblasts, are in the bone marrow. They're released immature. T cells go to the thymus, and B cells go to the bone marrow. Okay. Then these clonally expand, and you get antibody-studded B cells. IG is immune globulin or immunoglobulin, which is an antibody. And then you get T cell surface receptors on T cells, and then they migrate to lymphoid organs, and they wait. They wait until they're stimulated by some type of binding with an antigen, by a T helper cell, and then they'll expand and they'll mount it. In clonal selection, you have to be stimulated by an antigen. So a pathogen has to hit the body, an antigen on the pathogen to stimulate the T helper cell, and it can stimulate the B helper cell as well. Okay, and then specific lymphocytes, that's B cells and T cells, recognize the antigen. Okay, and they're already pre-programmed. That's one of the things that students have a hard time grasping, is that all of the immune cells that you need for any antigen that would penetrate your defenses are already in your lymphoid organ. Okay, you just have to select the right one, and all the acquired immune system does is it selects the right B cells and T cells in order to fight off that particular pathogen based on the presence of the antigen. Okay. These lymphocytes, once they see the antigen, then they go through a really rapid mitotic division, and they mount an army of the same specificity. So you get a lot of B cells and a lot of T cells that are all specific to one type of antigen. Okay. And like I said, lymphocyte specificity is pre-programmed. So everything that you need in order for recognizing antigens, you have at birth. It's just that those lines have to be stimulated, and then you have to get, in order to get acquired immunity, you get memory B cells and memory T cells. And without those memory B cells and T cells in place, then you will not have immunity. So you have to be stimulated first by an antigen in order to develop memory B cells. Okay. Now, each genetically distinct lymphocyte is the gene shuffle within B cells and T cells. So they will only produce one type of lymphocyte, and it only expresses a single specificity. So you only get a single type of antibody, or you only get a single type of T cell receptor. Clones that react to self, we don't want that to happen. We don't want antibodies to start attacking our kidneys. Those are deleted. That's a part of the role of major histocompatibility complex 1. It is self. And so when you have a clone that reacts to self, it's automatically deleted, and that's taken out of your immune repertoire. Okay. Now, if some of these forbidden clones are not deleted, then that leads to autoimmunity. So if you have clones that are floating around that will attack your pancreas, or attack your kidneys, or your liver, your spleen, your skin, whatever, then and you lose that immune tolerance, it's because forbidden clones have not been deleted, and they have survived and will then start to attack your tissue. Okay. So when we have an expansion, as we start out with stem cells, okay, then you get expansion of all these unique clones. Okay, these are B cells and T cells, and they all have different types of receptors on them, and they're all specific for different antigens. When these handshake with self, then they're deleted. Okay, so if you get a reaction with major histocompatibility complex 1 and they're interacting with self, then these forbidden clones are eliminated. So then what exists and what survives are only clones of B cells and T cells that are for antigens that are non-self. Okay. Then once these are selected, once this antigen drifts in and selects this particular clone, then this clone will rapidly divide. Okay. And then it will raise an immune response that is specific to this antigen only. The rest of these just lie in wait. They're just part of the army that's waiting. This one is selected, and so it will form a cloned army, just like Boba Fett in Star Wars. But you form a cloned army here, and that cloned army will go after this antigen and this antigen. Okay, I think I will stop there for our first part of this video lecture.